to all the judges. I'm Vivien. Today, my team and I will be presenting the project Inhibition of P.gingivalis with dual promoter DNA vaccine towards the prevention of Alzheimer's disease in mice model. Alzheimer's disease is the most common cause of dementia, which could go up to 75%. AD is characterized by the changes in the brain due to the buildup of abnormal amyloid plaques and neurofibrillary tangles. Globally, the number of people with dementia has been increasingly steadily since 2013, and this number is estimated to hit 152.8 million in 2050. Alzheimer is not just memory loss, Alzheimer kills. From this figure, we could see that AD is the top 7 leading cause of death in the United States. Next, I would like to introduce the Phosphoromonas gingivalis. It is a gram-negative gum disease-causing bacteria closely related to AD. It will produce the arginine-specific cysteine proteinases and lysine-specific cysteine proteinases. Both of them play a crucial role as a virulence factor from P.gingivalis in the development of periodontal disease.
Greetings to fellow judges. My name is Hui Long. So, how are the bacteria associated with Alzheimer's? P.gingivalis secretes molecules named gingipanes with variants such as gingipane A and K. The proteins can cleave off extracellular matrix components such as occludin in the blood-brain barrier and various immune response components that would help them evade the immune system. P.gingivalis first propagates from the periodontal ligament, and through the nerves, they get to reach the locus coeruleus and finally reach the hippocampus, which is part of the brain that is responsible for memory. In the context of amyloid beta, gingipanes can cleave amyloid beta precursor protein and form amyloid beta plaques, whereas in the context of tau protein, gingipanes would trigger the production of kinases and would hyperphosphorylate tau proteins, making them to be insoluble and aggregated, and neuron signals will be impaired.
Good day to all judges. I'm Yi. Now I would like to explain how the dual promoters work. Dual promoters have two types of expression system. One is prokaryotic expression system and another one is eukaryotic expression system. In the prokaryotic expression system, plasmid can be produced by using E. coli. In the eukaryotic expression system, it is usually used for expression. When it enters into the human body, the RGBA KGP will be expressed in the animal models when the eukaryotic expression system is activated.
Since gingipanes play a crucial role in its pathogenesis and is greatly associated with Alzheimer's, we decide to synthesize a vaccine by cloning the RGPA KGP proteinase complex gene in a plasmid, and it will be housed by an attenuated Vibrio cholera VCU-SM14P bacteria. The bacteria has subunit B on its cell surface, and this subunit acts as an adjuvant. Subsequent mucosal immune response is induced when the RGPA KGP protein is expressed by BCU-SM14P bacteria through the prokaryotic promoter. Once the BCU-SM14P is taken out by the M cells in the intestine, the plasmid's eukaryotic promoter is expressed and systemic immune response is induced. This slide shows the summary of how the systemic immunity against P.gingivalis is stimulated. The Vibrio cholera bacteria is first recognized by immune cells and internalized. The bacteria will then fuse with lysosome and it will lyse, and the plasmids will be released. In the end, the plasmids will be engulfed by dendritic cells, and the gene is expressed as protein presented by class 1 or 2 MHC and delivered to CD4 plus or CD8 plus T cells.
This flowchart is showing the vaccine development phases of our project. The first phase will be focusing on the development of the DNA vaccine. The second phase will be focusing on the animal trial, while the third phase will be focusing on the toxicological studies and preclinical trials. As for the mice study, the experiment design will involve two groups, which is the test group and the control group, where the mice involved will have Alzheimer's disease. And this is how our workflow will look like. At this point, I would like to highlight the Morris water maze mechanism. So in Morris water maze, the platform is hidden and placed in the middle. The mice will have 60 seconds to find the platform. Before the actual trial, the mice will be trained so that they remember the way to the platform. For the mice that don't have AD, they are able to find the platform. However, under the same circumstances, the mice with AD will not remember the way. And this is how we test our efficacy of our vaccine in the mice model.
Next, I will be continuing why this vaccine is novel. The bacterial vector used is called VCUSM14P. It's a live attenuated Vibrio cholerae strain that has been proven to be non-toxigenic with enhanced colonizations and immunogenic properties against V. cholera 0139 and cross-protective against enterotoxigenic Escherichia coli ETEC. The virulent genes of Vibrio cholerae are removed before insert into the animal models. Once the animal models receive vaccine, the immunity will be protected against cholera, enterotoxigenic Escherichia coli ETEC, and Alzheimer's. The vaccine platform is cold chain free universal bacterial vector UBV DNA vaccine for Alzheimer's disease. And its stability, because DNA is more stable than RNA vaccine when delivered in a bacterial vector. Next, cost effective. In single dose, it can protect against 3 types of bacteria or diseases. Next, safe and effective. VCUSM14P proven to be non-toxigenic, highly immunogenic, and protective in animal models. In conclusion, administrations of the vaccine may result in a reduction in the level of colonization by P. gingivalis. Thus, it can prevent or reduce the chance of infection by P. gingivalis. Last but not least, oral hygiene is also a very important factor associated with Alzheimer's disease. This is our reference. That's all from our groups. Thank you.
Thank you.
Thank you.